Modern technological tools like eDNA analysis and global databases are revolutionizing biodiversity conservation. They are enabling scientists to track species in inaccessible regions and study long-term trends. These innovations offer comprehensive insights into ecosystems, helping to predict future challenges and guide conservation efforts more effectively than traditional methods. Biodiversity conservation has entered a new era. Apart from relying on catching animals, counting plants, or direct observation, scientists are now focusing on modern tools from environmental DNA analysis to decades-long specimen archives and global species trait databases and tree ring analysis. These approaches are opening new windows into the natural world and making it easier to study regions that are otherwise inaccessible. In the far northwest of China, the Kashi River flows through a remote part of the Ili River Basin. The region is rich in biodiversity but difficult to study due to harsh terrain, seasonal ice cover, and limited resources for regular surveys. To overcome this, researchers turned to eDNA metabarcoding, a method that collects water samples and identifies DNA traces shed by fish. They collected 37 samples in 2 seasons, April and September, from 21 sites, spanning upstream and downstream stretches of the river. The DNA was then filtered, sequenced, and matched to known fish species using bioinformatics tools. Some 166 fish species were detected compared to just 27 found in earlier net surveys. While eDNA reveals what is living in a habitat today, other scientists are using archived natural materials to look back in time. In Germany, the Environmental Specimen Bank has been storing samples like tree leaves, bladderwrack seaweed, blue mussels, and zebra mussels since 1985. These organisms act as natural DNA samplers, passively collecting genetic material from their surroundings. Researchers from Trier University analyzed 550 samples collected over nearly 4 decades from terrestrial, marine, and freshwater sites. Scientists are also applying advanced global species trait and threat databases to assess the conservation status of turtles and tortoises, ancient reptiles that have survived for millions of years but are now facing unprecedented challenges. Researchers compiled data on 378 species from the IUCN Red List, specialist databases, and regional records. They included information on traits such as body size, geographic range, and reproductive patterns, along with data on threats like poaching, habitat loss, pollution, and climate change. Statistical models revealed that turtles and tortoises in South and Southeast Asia are in serious trouble, with about 85% of species at risk. The studies are not limited to animals. Using global tree ring datasets, scientists estimated the maximum ages of tree species and linked these to climate conditions during their early growth years. They examined how growth rates, species identity, and environmental factors affected longevity. The findings showed that trees experiencing mild climate stress in their youth often lived longer, and older trees displayed remarkable resilience to environmental changes. Although DNA has long been a tool in conservation science, these studies push its use to an entirely new level, placing them at the forefront of a new generation of biodiversity research. In the Kashi River project, eDNA was not used as a one-time survey tool, but applied across 2 seasons and 21 sites, revealing both seasonal and spatial changes in fish communities. The archived specimen research in Germany turned decades of stored natural materials into time machines, producing multi-decade biodiversity trends. The global turtle and tortoise assessment went beyond mapping current threats by combining species traits, evolutionary relationships, and human pressures to forecast risks for unevaluated species. Together, these studies show how modern tools are moving from simply detecting species to building connected, multi-scale pictures of biodiversity from past to present to predicted futures. This integration of eDNA, long-term archives, big data modeling, and climate-linked trait analysis marks a shift from isolated observations to powerful predictive systems. Modern technological advancements are transforming biodiversity conservation, enabling scientists to track species in remote areas and study long-term trends, marking a new era in biodiversity science.